So why do we perform biomedical research? Well, we perform biomedical research to try and make people healthy, keep them from getting sick. But to be able to make people healthy, keep them from getting sick, we have to understand why they get sick, how they get sick, and how we can intervene to prevent that. And to do that, we actually have to know the mechanisms that are involved. We have to know how the cells work together. We have to know how the proteins work together. We have to know what happens when a virus or a bacteria gets in there and messes things up. To do that, we have to have a system where we can manipulate things and understand how they change and how we can intervene. And that's where mice come along. We use mouse models because mice actually are a great model. We have the ability with mice to change things in them. Genetically, we can make them different. So they all look different, and we can make little changes and see what happens then with the mouse when it gets infected or whatever happens with making the mouse sick. We also have lots of tools that allow us to play with things in the mouse, and probably one of the best parts about mice is that they reproduce very quickly. That gives us a lot of mice to work with, and we can make a lot of changes quickly. Now, I know, we are not mice. Mice are not humans. But mouse models allow us to make these changes and allow us to see what these pathways are. The mice don't have the disease. The mice really are just a tool. And whatever we find in the mice, we have to take and look in the human to see whether or not that is actually operating in the human and whether we can intervene or not in the human. Let me make this a little more concrete for you and tell you about our work in asthma. So what is asthma? Well, asthma is a disease where people have a problem getting air out of their lungs. So on the left side is a normal airway. Wide open, air goes in and out, no problems. On the right side is the airway of someone with asthma. More smooth muscle, it's narrowed down, there's mucus production. All of that is impairing the ability to move air in and out of their lungs. So that's asthma. Why do we care about it? Well, it affects about 300 million people in the world. That's about 8% of the US population that has asthma. It costs about $82 billion annually. We're in a pediatric hospital. It's the leading chronic disease of kids, and it leads to about 14 million missed school days every year. And probably most frighteningly of all, asthma kills 10 Americans every day. Now, one of the things about asthma is we don't know why people get it. But there's a risk of developing asthma if you have a severe viral infection early in life. You may have heard of respiratory syncytial virus, or RSV. Infection with that virus early in life as an infant greatly increases your risk of developing asthma. So we wanted to understand what are the mechanisms that tie the viral infection to the development of asthma. And for that, we turned to a mouse model. Now, we used Sendai virus. Sendai virus is a virus that's like respiratory syncytial virus, but it's actually a natural rodent pathogen. That means it normally will infect mice in the wild. And in fact, in the wild, this virus can actually kill mice. But used at the amount we use in a mouse, the mouse will get infected, and it produces something called IgE. IgE is an antibody. It's a protein that people with allergies and with asthma also make. So it's kind of interesting that the mouse does that too. But more importantly, the mouse develops bronchoconstriction and mucus production. The same signs and symptoms you have of asthma in a human. Now, the mouse doesn't have asthma. The mouse is a model that allows us to look at the mechanisms that lead from the Sendai viral infection to the development of bronchoconstriction and mucus production. Now, the interesting thing we found when we looked at this model was we didn't see exactly what we thought we would see. So normally, when you have a viral infection in the lungs, it's kind of like an invading army. So we have our Sendai virus coming in there, and it's going to attack a certain cell in the body called an epithelial cell in the lung. And when the Sendai virus infects that epithelial cell is going to call on that radio for the immune system to come in and get rid of the virus. And the T cells and antibodies come in and get rid of the virus. And everything's good, and you go about your life. But what we found with the mice is that, interestingly, the virus invaded just as normal. It infected an epithelial cell, just like It would, what we expected, but this epithelial cell didn't call on its radio. Instead, this epithelial cell screamed these things called leukotrienes. The name doesn't matter. The point of it is it was different. And these leukotrienes brought in these cells called neutrophils, and in fact, a very specific type of neutrophil, not those other T cells and antibodies. And then these neutrophils ran and hugged a cell in the lung called a dendritic cell. And the interesting thing is that when they hugged this dendritic cell, they made this dendritic cell, or these dendritic cells, able to detect where the virus is in the lung, sort of like turning on a radar system. When that happened, the dendritic cell then called in another part of the immune system by using a thing called chemokines. Again, the name doesn't really matter, but that brought in a new set of T cells, sort of like these jet fighters. And these T cells were loaded with things called cytokines. And they went there and they used their cytokines to destroy the virus. Okay, great. Unfortunately, these cytokines also caused collateral damage. They hurt the lung, and they led to the development of the bronchoconstriction and mucus production, or asthma. So we found a new pathway, and many of these things some of these things were known, but many of these things were new, and in particular, the epithelial leukotriene neutrophil dendritic cell link was not known until we had done this. And it's interesting because this now gives us potential targets that we could go after to try and prevent a viral infection leading into the development of asthma. Now, as I said, this is in a mouse model. It's not humans. We have to verify if these pathways exist in the human, and indeed we are doing that and finding that most of these are in the human. And then we can look to try and make these into therapeutics to try and prevent the development of asthma. So what I ask you is the next time you're sick and you take a medicine, or you go to a doctor, you get a diagnosis, they can tell you how the disease works, why you're getting the disease, and what you can do to intervene and prevent it, I want you to thank your rodent friends, because they're working to keep us healthy. Thank you.